Hi, I'm Steffi, and in this video I would like to explain you my PhD project called Functional Dissection of Regulatory T-cell Plasticity in Health. The immune system consists of an army of various cells to protect our body from harm. When a pathogen enters the body, they will recognize this and start up an immune response to clear the danger. This immune response, however, needs to be well controlled to prevent unwanted response. Regulatory T cells are specialized immune cells whose job it is to suppress the effect of other immune cells, like, for example, effector T cells. Scientists used to believe that these cells were a very stable population. However, in the last few years, research has shown that some of these regulatory T cells can sometimes lose their suppressive function and gain functions of effector T cells. This phenomenon is called regulatory T cell plasticity, and these regulatory T cells are referred to as ex-Treg. In my project, I would like to understand why this phenomenon occurs and what the fate of these ex-Treg is. Plasticity is hypothesized to be important for an adequate immune response. However, when not well controlled, ex-Treg might actually help cause autoimmune disease, like, for example, diabetes and arthritis. Also in cancer, plasticity can play an important role. Tumor cells might prevent regulatory T cells from becoming ex-Treg in order to protect themselves from the immune system. Learning how to manipulate and control plasticity is thus important for many diseases. As the immune system is an intricate interplay between cells in different organs, I am developing a mouse model to study this phenomenon. With this mouse model, I am trying to visualize the different plastic cell populations with a technique called fate mapping. With this technique, I will track the expression of a gene specific for that cell type, in this case, regulatory T cells. 2 labels will be assigned to follow the fate of this cell. One label, called the life reporter, is to follow the life expression of this gene, and is shown in blue. This reporter will only be on when the regulatory T cell is actually expressing this gene and is thus a true regulatory T cell. The other label is called the fate mapper, here shown in green, and will also be turned on upon gene expression. This label will, however, once turned on, remain with the cell throughout its life. When a regulatory T cell becomes an ex-Treg, it will stop expressing the blue life reporter, but it will keep the expression of the green fate mapper. As such, this can tell us that this cell was once a regulatory T cell. When a cell gains effector T cell function, it will also turn on these labels, but in different colors. In this case, for effector T cells, the life reporter is purple and the fate mapper is red. This model also allows us to track plasticity from effector T cells into regulatory T cells. Effector T cells will express the purple life reporter and red fate mapper. When becoming a regulatory T cell, it will lose the purple life reporter but retain the red fate mapper. Also, as this induced Treg has become a regulatory T cell, it will turn on the blue life reporter and green fate mapper of the Treg identity. This mouse model will allow us to track all these different populations in a life situation. When a pathogen enters, some regulatory T cell, for example, will become ex-Treg, which we will now be able to visualize. As such, we can better understand the importance of plasticity in an immune response. To conclude, this model will allow us to understand plasticity between regulatory T cells and effector T cells. Further, we can better study each individual population to dissect the molecular mechanisms that drive plasticity. And lastly, we can study the effect of disease on plasticity. This will allow us to understand how we can guide the immune response and develop better therapies. Thank you for watching this video, and I hope I could give you a better insight in my PhD project. See you next time!